Okay, for the last section of our discussion on AMI, we're going to look at how the cells actually interact with one another, and then what happens to those interactions, how those interactions influence what's going on in the cellular response. Now, when we looked at this before, if you recall, we looked at an interaction between a T cell and an APC, and then we looked at all the individual interactions that we could see here, CD28, B7, T cell receptor, MHC2, all these things that then result in processes, cells being activated to produce cytokines and do other things. In practice, however, these interactions actually comprise an interface between the two cells. And if we look here at this top part here, if we look at here, we can see a cell. This bottom part would be the T cell here, T cell, and then the cell that it's interacting with would be interacting with it. And you can see there's basically those receptors and those interactions that we kind of turn it on its side. We can see that here are all kind of dispersed. There's a dispersed of the interactions at the T cell receptor, CD28, etc. Now that is the first event. That happens in the first 1 to 2 minutes. There is then a process in which the interactions between the cells are contracted or come into a more firm sort of type of organization, and you see microclusters basically, in which the blue here is the T cell receptor. T cell receptor is being clustered into the center of this set of interaction between the two cells. This matures for 10 minutes to several hours, in which you actually see this very distinct organization, in which the T cell receptor, the CD28, and adapter proteins and things are all clustered in one to provide a very large number of the T cell receptors in the center of this what we refer to as like a bullseye. And then there's different ranges. There are different proteins that are in these ranges around that surround this area. Now we can see that a little bit clearer here. If we look on the side, the interactions would have the T cell receptor and the MHC2 on the APC side would be clustered in this area, and this is referred to as the immune synapse. And so just as you think about, there is a synapse between neurons, between the axon terminals and the dendrites on a neuron, two neurons. You can see here that there's a cluster effect in which there's a distinct organization that happens over time of those receptor and ligand interactions. Within the center of this in this center bullseye type, and you can see up here there's red is basically this ICAM, which ICAM is ICAM LFA1 interactions are you can see in red that surround then the T cell MHC2 interactions, and they form this bullseye, and they're distinct proteins, distinct types of proteins that interact in that bullseye. Well, in the center you have the T cell receptor and the MHC2 plus CD28 makes sense, and the B7. All of these on the APC side, and all of these have maximum interactions here. This serves to really enforce the interactions between the cells and to really amplify that response between them, because you have multiple receptors being activated at the same time. One thing I want to point out is there are these LFA1 and ICAM. LFA1 on the T cell, ICAM1 on the APC, is in the outer rings of this bullseye. And if you remember what I showed you before, I told you before, these are referred to as adhesion proteins. And you can see here they basically corral that T cell receptor MHC2 in the center, and they form basically this almost like molecular Velcro that keeps the cells together. They do have some signaling properties, particularly LFA, but for the most part, they really keep things. They keep things in an organization by organization around this circular organization around the T cell receptor and MHC2 to really enforce that interaction. Now we can see this again here, the immune synapse in this electron micrograph, in which if we look at a T cell, if we were to strip away, if we sort of take away the APC, we can see the green here is stained. That's a stain that the cells have been stained with, and basically an antibody that stains in. That's where the T cell receptor CD4 and CD28 reside, all in here. And then the LFA1 ICAM interactions. LFA1 is going to be all in this area, this red part, forming this bullseye-like interaction. And so there's distinct, what I want you to understand is there's distinct organization of these, with the LFA1 adhesion molecules on the outside, and in the center of the bullseye, the synapse, you have the T cell interactions, B7, MHC2 interactions, B7, CD28 interactions, CD4, all happening in that center. Now the thought is that this really reinforces again that interaction and the signaling that needs to go through to the cell. Here's another it doesn't look like it's working, or it's very slow. Just to give you it's another set of interactions, just to show you how the T cell, here we go, how the T cell interacts. You can see right there, you can see the synapse, and you can see it right there, that interface between the T cell and the APC. Let's take another look. There's another one. There it is again. And so that shows you there's a really distinct set of organizations between them. And here in another type of staining, you can really see that interaction between the cells, which in this case you can see this nice bullseye between the cells. And so again, there's a really structure to the interaction between the two cells. It's just not single interactions between them. Adhesion ring, and then the signaling zone, and then a secretory zone. Now I'm going to skip over this because we'll come back to this later because this will involve another type of cell, CD28 cells, CD8 cells, sorry. So what's happening in that signaling into the cell? Well, there's a set of proteins that are involved in this signaling, and we're going to look at those. Those proteins, what I've given you here is a little glossary of the different proteins that are important for cell signaling. Phospholipase C, diacylglycerol, protein kinase C, nuclear factor NFAT, or nuclear factor of activating T cells. These regulate IL-2 expression, AP1, which is an activator regulating of cytokines, including several cytokines, including IL-2 production. NF-kappa B regulates additional ones as well, and I-kappa B is an inhibitor of kappa B. So I know that doesn't mean really much to you right now, but I just want to show you, because we're now going to look at that mechanism. You can refer back to this little glossary. So if we look at what happens in the cell, so if we look at the interaction now between, the cells here. Look at that interaction here. The T cell receptor, the MHC2 peptide, and CD4, and then we have these interactions with the with CD3. CD3 turns out is made up of several subunits: the epsilon, delta, gamma, and zeta, and those really form this T cell receptor total complex. This is one of the largest receptor complexes actually that we know of, and for good. And it has a lot. There's a lot of functionality here. Most importantly, the zeta, because it really, you can see it sticks right directly into the plasma, um, into the cytoplasm, serves as a place where when you get these events happening, results in the attraction of a kinase, LC kinase, this LCK kinase. This kinase will bind to the CD4 site when this complex is formed. This this kinase then phosphorylates the zeta chain of the T cell receptor to CD3. This results in the binding of another kinase of ZAP70, which, when these zeta chains are phosphorylated, provides a landing pad for ZAP70 to bind to the T cell receptor. When it does this, it can now undergo. It is encouraged to undergo ATP hydrolysis to phosphorylate phospholipase C. Now, phospholipase C is at the membrane. It has, and being at the membrane, what it does is it hydrolyzes inositol triphosphate, inositol triphosphate diacylglycerol, which is which is in the membrane. It's a type of phospholipid that's in the membrane. That's a signaling molecule because it's referred to as a secondary signaling molecule, and it produces diacylglycerol, which I have up here, and IP3 here, inositol triphosphate. These molecules, it basically cleaves that. It cleaves basically the triphosphate off, and forming diacylglycerol, which will stay in the membrane, and IP3. Now these are two events. This is sort of a bifurcation of the signaling, because the IP3 is important because it results in, it goes and basically acts on intracellular calcium stores. There are calcium levels in the cell is very extremely low. Outside the cell, it's very high. There's over 20 or 30 times more calcium outside the cell than inside the cell. But there's actually calcium within the cell in stores. It's stored. It's not available to the cytoplasm. IP3 interacts with those, and it's the interaction with these calcium stores results in the release of intracellular calcium. Now this is important because all things are basically being driven by this intracellular calcium activation. Now this binds to a molecule called calmodulin, which is specifically set up to bind to calcium. As the calcium levels rise, more and more of the calmodulin becomes bound with calcium. And then as this concentration rises, it now binds to another molecule called calcineurin. Calcineurin forming this complex of calcineurin, calmodulin, and calmodulin. Now this is activated. This is now an active molecule, and instead of doing a kinase reaction, what it does is it acts on a transcription factor NFA2 phosphate and converts and actually cuts this off. So it removes this phosphate. It acts as a phosphatase, and as a consequence, in dephosphorylating NFAT can now migrate into the nucleus. NFAT is a transcription factor, and in its phosphorylated form, it spends most of all, if not all of its time in the cytoplasm. But when the phosphate is actually removed, and you have NFAT in the cytoplasm, it now wants to spend most of its time in the nucleus. It can go now and bind as a transcription factor to the chromosomal DNA in the promoters there, making a complex, in particular with another transcription factor, AP1. Now, let's just go back a second here. So, now, Okay, so that's one story. So again, all of this that we went through, phosphorylation, results of IP3, calcium store activation, activation of the transcription factor, is to get these transcription factor complexes on the chromosomal DNA. Because what's going to happen is we're going to get this will and they encourage the transcription of the genes that they need to transcribe, for example, cytokines, as potentially other proteins. Now let's go back up here at the top where we have the NAG up here. So NAG, it just doesn't hang out there. It has a role to play, and that role is to interact with protein kinase C. And it's a stimulator of protein kinase C activity. And when it does that, when that concentration of DAG goes up, it can activate the protein kinase C. And what this does is it causes it causes the phosphorylation of NF-kappa B, or the inhibitor I-kappa B. And in this case, in phosphorylating the I-kappa B, which is in a complex with NF-kappa B. So let me just step back here a minute. This transcription factor is held in the cytoplasm. It's basically inhibited because it has an inhibitor that's bound to it, inhibitor of kappa B, I-kappa B. And when protein kinase C does is it phosphorylates the I-kappa B, and this releases it from the NF-kappa B. And that releases, that's a little bit simpler than what we were talking about before with NF-AT. The NF-kappa B can now migrate into the cellular membrane. And it can then also bind to the forming a complex with these transcription factors, leading to transcription of IL-2 and other cytokines. Particularly IL-2 is important during this activation process. Now, so what you have here is the activation of backup NF-AT involves dephosphorylation of the phosphorylated NF-AT, and the activation of this transcription involves phosphorylation of its inhibitor, releasing it so it can go it can migrate into the nucleus and bind to its promoter sites for the cytokines it's supposed to activate. So this is really important. This sort of scheme is important because, as we'll see later in the course, there are a number of drugs that act on different spots in this to modulate the activation of T cells. It's important to know this also because this is the way in which T cells go through their activation. This is how the signal from all the way that antigen that was taken up by the antigen processing cells gets translated all the way to activation of the T cell, which in this case a CD4 cell, which is going to go on, as you recall, and produce memory cells and more of these activated T cells. So this is the language that we have. This is the language in which is written and how these signals are communicated from antigen specificity bound and from antigen interactions to the generation of T cells which have specificity for a particular antigen. So the last thing I want to talk about is what's referred to as T-independent AMI. And this means that there are no T cells that are actually involved with this in the initial spot, the initial interaction, as we'll see. It's characterized by a number of things. One is it was observed that you could get a response if you had T-deficient animals, that you could get this kind of response, and so it didn't necessarily involve T cells. Lower or low affinity antibodies are generated directly without the need of T cell activation. And most T-independent antigens have repeating subunits that are arrayed in regular geometrical shapes. So a T-independent antigen has a distinctive geometrical shape and size, and it's repeating repeating subunits, and many have high molecular weight, like high molecular weight polysaccharides, LPS, dextrans sulfate. These can all result in this mechanism. Most importantly, virus capsids are excellent inducers of strong T-independent AMI, and this is one of the first events for viruses, non-envelope-based viruses, is one of the first events and one of our strongest parts of the immune system antiviral responses that we have. If the antigen processing takes place, which it will do, as we'll see later on after the initial event, then isotype switching will take place and high affinity antibodies will be induced. So how does this work? So if you have a B cell, for example, and this occurs with B cells, this T-independent AMI in the initial stages doesn't involve macrophages or dendritic cells, and it comes into contact with a antigen which has repeating subunits. So each one of these red balls is the same antigen determinant, one right after the other along this repeating subunit, one right after the other, in distinctive distances, somewhere around 30 to 50 nanometers. So it's one after the other. So this is unlike a lot of antigens, which like for proteins, which may have just that one antigen determinant on it. These type of antigens have repeating antigen determinants. They're exactly the same, one right after the other, like a string of pearls, basically. You might think about it. Now, in that interaction, what happens is means that they can bind to multiple antibodies on the surface of a B cell, and therefore lead to clustering of those B cells, clustering of the antibody on the B cells. What this induces is a very simple proliferation of the B cells. You'll make more of those B cells. However, because T cells aren't involved, there's no memory cells that are kind of generated from this interaction. And the first set of what you get out of this is high levels of IgM are produced. So there's proliferation, modest proliferation, no memory. In other words, there are copies of the B cell that are made that produce the antibody, and then these B cells can produce and excrete IgM. So if we look at this, say from a virus, if we have a non-envelope virus which has repeating subunits all around its capsid, you can see if you have a naive cell, it can bind to those, and oligomerize the antibody on the surface of a B cell. This will initiate initiates IgM production. So you'll get some strong IgM production from the B cell. And then what can then happen is if the virus is taken up into the cell, then then what can happen is antigen processing, which you already know about. You know about this. Excuse me. Antigen processing will occur, and then what you already know, B cell activation can occur, and IgG production can occur. So the point here is that you have IgM production in this initial step, but once the virus is taken up, or a particle is taken up that has these repeating subunits, and it's a protein, then you can also get what you already know about antigen activation, regular AMI-based response. So I just want to point out is that the T-independent part of this is this part here, all this part here. I saw it before. This part here, down here, is going to be T-dependent, right? This is going to need T cells here. Okay. So how does this all fit together? So as you can see here, you have virus. It can be taken up by a macrophage, And we see here on the side, let's put my pen here, it can be taken up by a macrophage or dendritic cell. As a consequence, you'll get a T cell activation here. The virus can also interact with a B cell, and it can for a brief period of time generate IgM production from that B cell. But if that virus is taken up into the cell, you can then also get antigen processing occurring. And as a consequence, then you can see how this would then lead to the T cell activation, B cell activation, B cell activation along at this point, and the production of plasmacytes and antibody, particularly IgG, to the virus. The virus can activated B cells once they interact, activated B cells, which are prone to really wanting to produce IgG, are going to interact with the virus, take it in. If this has occurred, go through antigen processing, and once again produce antibodies and memory. And in this case, what you're going to get is you're going to get memory cells. Memory cells are going to be produced, and plasmacytes are going to be produced. So antibody will be produced as well as memory cells. So in this way, you can get an initial response. You get the first event you would get this IgM production. Put that number 1 here. And then after you go through what you already know about the AMI production, you're going to get IgG, eventually IgG production. And so that gives you an initial protection and then long-term protection by producing antibody and memory T cells, as well as memory B cells, as well as memory T cells. So to wrap up, for the antibody-mediated immunity take-home message, what have we looked at? Well, first, that the generation of antibodies to an antigen depends on the initial and secondary exposure to the antigen. You've seen that. You know how important clonal selection now is and somatic mutation are to governing this production of these tight antibodies. CD4 positive T cells control this B cell antibody selection and production, and that production also is other APCs like dendritic cells and macrophages are important for priming that system and getting that system going so that they can activate the B cells to get, in the end, memory B cells, memory T cells, antibody, and more activated T cells and B cells. And antigens can be used to induce antibody-based immunity to some microbial pathogens, as we've seen, particularly if those antibodies, if those antigens have regular repeating subunits, then you, like non-enveloped viruses, which then will produce basically a burst for a transient period of time of IgM, followed later if taken up by B cells and also by macrophages, into a strong AMI IgG response. Okay, that's what the end of what we're going to talk about now. And I will see you at our next lecture.